All right, so the discussion of beta receptors in the heart makes a nice segue into the next portion of our lecture series, and that's on the cardiovascular system on plate 31. You're going to spend some time in the laboratory talking about the anatomy of the cardiovascular system. You're going to dissect a heart. You're going to kind of review the blood flow. My experience has found that this is probably a great place for a little re-edification both in lecture and in lab, that the heart is a pump. It consists in our domestic species of 4 chambers. It has a divided blood flow. One portion of the blood tends to go to the lungs. That's referred to as the pulmonic circulation. The other portion of the blood flow goes to the rest of the body, what is referred to as the systemic circulation. So I will talk about pulmonic and systemic blood flow, recognizing that there are different tissues involved. And traditionally when anatomists draw pictures, they draw it as it lays in the animal. So the left heart is on the left side, the right is on the right side, as it would be lying in the animal. We have the atria, we have the ventricles, we have the connecting tubes. Systemic flow goes, I should probably do it. Let's do it here. Let's look first at anatomical flow. There's left ventricle, right ventricle, and then there's a left atrium and a right atrium. And again, you're going to go over this in infinite detail, hands-on in a laboratory cutting up hearts. The blood flow from the left ventricle comes out the aorta and to the rest of the body, the systemic circulation. It then comes back into the right atrium and right ventricle. And out of the right ventricle, it goes to the lungs. So these are the lungs. So left heart, left side, systemic circulation, right heart, pulmonic. If you look then at the functional flow, here's the left heart. It's driving blood to the systemic circulation. That is returned back to the right heart, and the right heart then pushes blood through the lungs. Systemic circulation, pulmonic or pulmonary circulation. The total blood flow is identical through this system. And that's something that may not be appreciated on the anatomic, but if you draw it functionally, these are values for humans. What it's telling you is that there's 5 liters a minute being pumped out in this direction. Well, the amount that leaves the capillaries, unless a bullet comes in here and severs an artery, the amount of blood that's leaving these capillaries has got to be the same. The amount going out of the right heart to the lungs also has to be equal to the amount coming out, and they've got all to be equal. So this is steady state blood flow. You're not having a net loss of fluid. You're not having a net gain of fluid. They've got to be equal. So the first property of flow and velocity through the blood system is that through any cross section of the circulatory system, flow has to be equal. So if you look at the flow out of here, it's 5 liters a minute. If you look at the flow through the capillaries, it has to be 5 liters. And then when it's flowing through the veins, it's 5 liters. Arteries carry blood away from the heart. Veins bring blood back to the heart. Blood flow through the arteries has to equal the blood flow through the veins, but they have to be taken total. Okay? Blood flow equals blood velocity times the cross-sectional area. So that flow equals velocity times area. And by area we mean cross-sectional area. So think of 2 tubes that are connected. Large tube, smaller tube. Flow is going to be the same thing through here as it is going to be through here, right? Unless it's leaking out somewhere. Let's just say 5 liters a minute. So if the flows are similar, area is greater, what happens to the velocity here? Decreases. You'll see that in a stream. Narrow stream running very, very fast. What happens when it widens out? Slows down. So if I go from a small tube to a large tube, velocity decreases. Conversely, what happens when you put your finger over the end of a hose? So you've turned on the tap, and you're going from, in the case of the hose, you put the finger, you're going from a large cross-sectional area to a smaller cross-sectional area. What happens to the velocity of that water? It goes up. What happens to the flow? Nothing. Flow is the same. But what happens is it comes out faster, and that translates into a farther jet of stream. Also, if you get hit with it, I mean, you feel the velocity. Total cross-sectional area of the capillary bed is much greater than other vessels. Capillaries, by definition, are very small. Maybe on the order of microns in diameter. Arteries are big. But the distinction is that you can have one artery, like your aorta, that branches and provides blood to billions of capillaries. The net effect is that if you take the cross-sectional area of all of those capillaries, it's very, very large. And that's reflected in this diagram right here. Right here. Here's the schematic diagram. The heart is pumping blood out in this direction, 5 liters a minute. It then branches into the capillaries, and then, so these are arteries, and these are veins, bringing blood back to the heart. This graph down here represents cross-sectional area. So while the cross-sectional area of one capillary is minute, if you take all of the capillaries together, they have a huge cross-sectional area. So we're going to take our first principle. Flow is equal to velocity times cross-sectional area. And we're going to ask the question, well, what happens then to the velocity of blood flow when you reach the capillaries? And that's reflected in the curve right there. Largest cross-sectional area, lowest velocity. If you think about it, one of the things you want to do at the capillaries is exchange nutrients, exchange products. The best way to do that is to allow these cells to move very slowly. So in fact, if you try to watch the velocity of blood flowing through an artery, very, very, very fast. But what happens is, in individual capillary beds, the flow is decreased by an order of magnitude. So the capillary then flows along with the fluid very slowly through the blood vessels, allowing for exchange to occur. For example, blood normally spends about 1 second in a capillary. If it was traveling at the same speed in an aorta as it was in the aorta, it would be through, instead of 1 second, it would be 0.001. That is a factor of 1,000-fold. So by having this large cross-sectional area, you have 1,000-fold more time to spend in exchanging material. On this plate 31, it also talks about oxygenated blood and deoxygenated blood. So we need to kind of also consider that. I remember getting a test question that they talk about, arteries always carry oxygenated blood. Well, that is true. Arteries do carry oxygenated blood for the systemic circulation. If we go back and we look at our functional anatomic distribution, here's the left heart. Blood is going in this direction, so that makes these arteries. There's the capillaries, that makes these veins. This blood is oxygenated. Oxygen is pulled out at the tissue level. So this then becomes deoxygenated. Goes to the right heart. From the right heart, blood flows to the lungs. So here's an example in which arteries are taking deoxygenated blood, and veins have oxygenated. So I make a distinction of arteries and veins based on where they're taking blood, not whether they have oxygen in them or not. Turn to plate 32, and we'll talk about the myocardium and the generation of an action potential. Soma, axon, synapse. We talked about neurons. We talked about neurons of the sympathetic and parasympathetic system. We talked about action potentials being propagated along skeletal muscle. So in response to an action potential from a motor neuron, you could measure an action potential traveling along the sarcolemma. What about cardiac tissue? That's an example of striated muscle. And in fact, it has a membrane which is electrically active. But what is really cool about the heart is it has an automatic or autonomic function. meaning when you cut the heart out, it will continue to beat autorhythmically on its own, without neural input, without any kind of hormones. It continues to beat. Now, in domestic animals, animals that have relatively warm body temperatures, this is not a prolonged period of time. A lot of the preliminary work on heart function was done on frogs and turtles. One, because of the demand for oxygen is about a tenth that of humans and other animals. Secondly, they can function at room temperature. You can have a turtle heart beat for hours outside of the body. We talk about striated muscle making up the muscle tissue. In lab, you're going to see a slight distinction between skeletal muscle and cardiac muscle. One of those distinctions is in the sarcolemma and this capacity for auto-regulation of the heartbeat. So we're going to talk now about the myocardium. The duration of the cardiac action potential is very long. We talked about action potentials in a neuron, 1 to 2 milliseconds. Very, very rapid. Now think about that. If you could get a contraction every millisecond, how fast would your heart beat? 1 millisecond, there are 1,000 milliseconds in a second. So, if your heart beat each time for each 1 millisecond action potential, that would be 1,000 times a second. That would be 60,000 times a minute. Action potentials last much longer in the heart. You're using that action potential to drive a contraction which has to pump blood. This is a much slower process. So instead of 1 millisecond, you may be talking about 200, 300 milliseconds. It is hundreds of times slower. Consequently, there's a long refractory period throughout the time of the contraction, the action potential. It's refractory, and it's absolutely refractory. Think about it from a functional standpoint. What if the heart contracted, and then before you allowed it to relax, that is, you shortened the refractory period, and you didn't allow it to relax, then it would contract again. In order for a heart to work, it must have a period of contraction and a period of relaxation, a reciprocal pump. That is, it has to contract to squeeze blood out, it has to relax to fill with blood. If you were to fuse the contraction, if you were to produce a tetanic stimulation like you do in skeletal muscle, the heart would be permanently contracted. Not a good idea. So what the heart does, one of the ways it protects against this, is have a very long action potential and a very long period of refractivity. So if we look up at this diagram up here, this represents an action potential as seen in the myocardium. So this is heart tissue, no nerves attached to it. The first obvious thing is that there's kind of this auto-depolarization. In a neuron, without any stimulus, we maintained an action potential that was minus 70, it was minus 70, forever. Now what we see in a myocardial cell is that there is a slow depolarization. In fact, there is a relatively high permeability to sodium, so that it continually depolarizes until it reaches a threshold. Like an action potential, there are voltage-gated sodium channels that open up. Sodium rushes in, goes from about minus 70 to plus 20, just like in an action potential, very fast. But now what happens is an incredibly slow period in which there is not a repolarization, there's more of a plateau. Same thing happens to the sodium channels in myocardial cells as it does in a neuron. When it becomes positive, that is, when it reaches this peak up here, the sodium channels are shut off. In an action potential, there tends to be an increase in potassium permeability so that there's a rapid repolarization. Well, in fact, in myocardia, there is some potassium leaking out, but more importantly, an increased permeability to calcium. So again, here's our cell. Here's our cell. High potassium, high sodium, high calcium. Slow depolarization. A little bit of sodium leaking in causes the inside to become a little bit more positive. When it reaches threshold, poof, big opening in sodium. That is the depolarization phase. When it becomes positive, when the inside of the cell becomes positive, that gets shut off. In the repolarization phase in a neuron, potassium goes out. Well, we see some of that in a myocardium, but more importantly, big increase in calcium. So even though you may have some slight positive charge leaking out, tending to make the inside more negative, you have it balanced by this calcium coming in. The net effect is very small changes in the membrane potential. So if we look at this in a little bit more detail, so there's that slow depolarization. Rapid depolarization associated with sodium. You see that? There's a little calcium coming out, making it a little more negative, but then it gets balanced. So instead of going down like this, it plateaus. It then reaches a period of time in which there is repolarization. Repolarization is associated with large increases in potassium, shutting off calcium. So by balancing the permeability and by having different concentrations on it, we can control this membrane potential. It's very long, maybe 200, 300 milliseconds, and it's absolutely refractory. And I think we'll break there.